Um, we welcome Cecili here. It's very nice to see her again. Cecili, as you know, worked in the laboratory of Svetlana Maslakova, so we will now turn the time over to Svetlana to introduce her.
Hello, everybody. I'm Svetlana Maslakova. Many of you know me. I'm a faculty member at the Oregon Institute of Biology. And I am very pleased to introduce Cecili Mendes, who I met first time as a participant in a Nemertian taxonomy workshop that I taught in 2018 in Panama at Bocas del Toro. Which is on the Caribbean side of Panama. And Cecili impressed me greatly with her enthusiasm and her maturity, so rarely observed at her early stage of career. About a year afterwards, I missed Cecili greatly, and she suddenly wrote and said that she would like to apply for a fellowship from the Brazilian government to come and do some work in my lab to learn how to raise Nemertian larvae. So we collaborated together to put together a proposal, and we proposed to do something that nobody had ever done before. We proposed to challenge a paradigm in Nemertian developmental biology that a particular group of ribbon worm larvae were in fact feeding in the plankton. Everybody thought they were not feeding at all. We had some preliminary data and some suspicions over the years, accumulated over the years, that they must be feeding because they are clearly growing in the plankton. So I suggested that Cecili come and tackle this black box in the life history of one of the classes of ribbon worms. And it wasn't clear that it was going to work at all because nobody had ever done this before. But Cecili came and she got the fellowship. She arrived on the first day. We went out on the intertidal, grabbed some worms, and it just all went amazingly well in Cecili's hands. Somehow, miraculously, the worms spawned immediately, and she was able to pick it up from the scratch. I was very busy, as always, not very attentive to her, but she worked independently, terrifically well, and was able to learn how to culture these larvae and learn how to feed these larvae. Turns out they like barnacles, which we kind of knew.
Don't tell spoilers.
Sorry. Well, anyway, so she was able to raise these things in the lab for almost a year and get them all the way to metamorphosis and to even sexual maturity, which was an amazing feat. While doing that, Cecili participated in all kinds of other projects, working with George even more than with myself. We miss Cecili greatly. She's a friend as well as a colleague. She's terrifically productive and just a wonderful person to have around. So I'm really glad to welcome Cecili to the seminar series and to hear about her work in Brazil on the genetics of marine worms and how it relates to their life histories.
Thank you, Svetlana. That was really nice. Might I start now? So I'm going to share my screen. And really, thank you. It was a really nice introduction. And as Svetlana told you, I'm gonna show a little bit of my PhD work here in Brazil. under the supervisor of Sonia Andrade. And I'm going to talk about the comparative genomics of 4 species of marine worms, 3 Nemerdians and 1 polychaete, with different development strategies. So, why are you not going? So first, just so you know what the structure of my talk, I'm going to start talking about what influences the population connectivity in marine populations. Then I'm going to characterize a little bit of the Brazilian coast for you that don't know much of my country, and how we conduct this study and the results that we found, that we have so far, and what these results indicate to us. Then I'm going to finish with a short summary and what we are planning to do next with this work. So first, it's very important for everyone to know that connectivity in marine populations is directly affected by the pelagic larval duration, PLD, which will be very important because most of these marine animals are benthic and they don't disperse much once they are adults. They disperse mostly when they are larvae, in the larval phase. And we can characterize these in 3 categories. The low dispersers are the ones that have larval duration of 1 to 15 days. The medium dispersers are the ones that have a pelagic duration of 16 to 30 days. And the high dispersers are the ones that have a pelagic duration of more than 31 days. And the life traits are also important, but mostly when they are adults, they will not disperse much. So when you have a low dispersal capacity, you have a high retention rate. So you have a higher differentiation in the genetics of populations, even the ones that are close geographically. You can see easily the genetic differentiation between these populations. When you have a species that has a longer larval period, you have intermediate retention rate, and then you can see the genetic difference being more acute in populations that are more distant geographically. And when you have a high dispersal, the one that stays for more than 31 days in the plankton, you have almost no retention. You have a low retention rate. So these populations are more well connected and they have smaller differentiation genetically. And these types of population also suffer different effects of the ocean currents and the barriers that can modify this geographic distance, because if you have such a low connection between these populations, you are not going to see much of the effect of currents and barriers because they don't disperse anyway, so it's not that important for them. But for the medium and higher dispersers, the currents and the barriers will have a greater importance because these are the ones that are going to shape how these populations are going to connect. And it's also interesting to notice that when you have low dispersal, these populations are very weak, are very vulnerable to environmental change because they'll probably, most of the time, they'll have a low diversity, genetic diversity. So in case of change in the environment, they'll have a small diversity to recover. And also, if you have some kind of local extinction, it's going to be hard for that place to be recognized by the other locations. And when you have medium and higher dispersers, you're going to have this larger genetic diversity, which will be better for them to recover in a changing environment. And also, if occurs extinction in a local context, this location can be recognized by populations nearby. Talking a little bit more about the contribution of the larva in this dispersal, here we have a graph showing the spatial context of these larvae. Each of these horizontal bars in the graph show the space that is occupied by these larvae, not necessarily the number of larvae. And once you have initial spawning output, the larva, the currents, the ocean currents will be the ones that will make these larvae disperse and make the space that they occupy larger. But of course, all the process that happened during this dispersal are also very important. So the larval behavior, the larva goes up and down in the water column, will be very important to shape how long a larva will disperse and how well it will disperse. Also the predation in the water column and the settlement, the habitat that it's available for these larvae once they finish their dispersal, and also the survival after they disperse, because even though they can find an available habitat to settle, they can be selected, they can suffer selective forces that will not let them survive, and this will also shape how these populations are connected. But the size of these populations also matters. It's also very important, because in general, of course, there are exceptions of this pattern, but in general, as a general rule, if you have a small population, which is here we have the effective size of a population and here the migrant rate. So if you have a small population with a low migration rate, you usually have a virtually closed population without much gene flow and very different genetically, even though they are close geographically. And when you have a big population with a high effective size and a low migration rate, is that when you see the geographic climbs, that's when you can see a change in a pattern happening in the distribution of a species. And this change usually occurs as a response to some kind of ecological and environmental variable. As when you have a median population with a median migration rate, you're probably going to see a stepping stone gene flow, which is more common for the invertebrates that we see in coastlines. When this happens, you have the close geographically population are also close genetically. So you have this similar, you can see isolation by distance when more close populations geographically are also close genetically. And when you have high migration rates, you are going to see either chaotic patchiness when you have a small population, which means you have different cohorts, genetically cohorts occurring in the same space. And or if you have a big population and high migration rate, you're going to see open populations, which are what we call palmitic populations, which is when you have almost the same population, the entire distribution of a species, because the rate of gene flow is so high that you cannot really tell when a population begins and another ends. Now, a little bit about Brazil. Brazil, it's the largest country in the South America. It's this blue patch in the map. And we have about 4,500 miles of shoreline. And in the north, around here, it's mostly sand beaches. We don't see much rock shores. Here in the northeast part, there are also a lot of sand beaches, but we also see some patches of rocky shore, mostly from sandstones. They are mostly like this. It's not really common, but they occur a lot. And in these rocky shores, we have these tide pools that are mostly shallow, but sometimes they can be about 2 meters deep. And these tide pools have a very complex community with many animals and algae occurring. In the southeast, we have what we call the true rocky shore, because it's the mountain Serra do Mar that goes through all southeast Brazil. And this mountain goes to the coastline, and you have the forest meeting the ocean. And in here, we have these communities, the sessile communities that occurs in the rocks, mostly oysters and barnacles and some mussels. And also when you have the low tide, you can see some tide pools in these areas here, most close to the ocean. And there's also many different species that occurs here. And what about the species that I studied? I use 4 species in my work, 3 nemerteins and 1 polychaete. It's a nereid polychaete. Actually, at the beginning, we thought we were working only with 3 species because we thought that the 2 Nematopsis species were the same. But once we started to look at these animals and see the genetic results, we realized that they are actually 2 different species, and we are now describing them. We also work with Heteronemerdium, the Linneo sanguineus, and with Perinere spontanei, which is a nereid polychaete. And we chose to work with these species because we thought they probably have different types of larvae and types of reproduction. So it would be very interesting to see what the population genetics tell us. About the species, the first species, it's a Nematopsis. It's a Haplonemerdian, and as Svetlana was telling you, these animals are presumably to have an abbreviated larval development, but we already have some genetic studies, especially with Autotyponemerdes, that shows that they probably have a long-lived larva, and also there's the difference in size of this larva in the plankton that points us to a probable long-lived larva. This species occurs from Northeast Brazil until Argentina, at least so far that's what we know, because it's a new species, so it might have a broader distribution. And they feed upon acorn barnacles. They live in those communities of oysters and barnacles and mussels, and we saw them feeding upon acorn barnacles, and we also recovered some gut content sequences from Tetraclita and Balanus barnacles. The second Nematopsis species, of course, is also a Haplonemerdian, and it's also a new species, so we only know that they occur from Florida to Southeast Brazil, but it might have a broader distribution that we don't know yet. And they also feed upon acorn barnacles, and we also recovered some sequences from the gut contents showing that they feed on Tetraclita and Balanus as well. There are some really common species here in the coast, these 2 barnacles. The third species, it's Linneo sanguineus, which is a Heteronemerdian. So we would think that they have a Palladium larva that it's common for Heteronemerdians. However, this species is known to have asexual reproduction. They break in pieces, and the pieces can rise a new individual. So they have clonal populations. This species is known to occur worldwide. As far as I know, they occur only in the Atlantic, in Europe, and North and South America, but they might occur in different places. They feed upon polychaetes. I've seen them feeding upon ciliate polychaetes. They might feed on other species as well, but this is what I saw so far. And the fourth species, it's a polychaete with a planktotrophic larvae. We never saw the larvae of this species, but we know they have a planktotrophic larvae because all the other species known from this genus has planktotrophic larvae that stays for a long time in the plankton. This species occurs from Northeast Mexico until South Brazil. And as far as we know, it's an omnivorous species that feeds upon anything that can find between Brachydontis and Zussus, where they usually live. So the sampling, we visited 15 places in Brazil from the northeast to south. These were four sites in northeast, 2 in the south, and the rest in the southeast, just because it was easier for us because we are here in Sao Paulo. So it was easier for us to go through the southeast. And from these places, we collect these communities of oysters and mussels here and some barnacles. We collect these animals, these communities, take to the lab, and there we trying to find our species of interest, because in the field we can not sometimes find them because they are pretty small. So Nematopsis species 1, we only found in 8 of these 15 sites, and we only found 19 specimens. Nematopsis species 2, we only find in 5 of these 15 sites and only 16 specimens, showing that these populations are really small. Linneo sanguineus, we found in 7 sites, mostly in the southeast and 1 site in the northeast. They are these pink squares. And Pairwise Fst, we found in 8 sites, and we found 61 specimens, both in the northeast, in the southeast, and we also found in the south, but we could not use the specimens from south because of the low quality DNA that we recovered. So we extract DNA from all these specimens, and we use a modified GBS protocol, which is genotyping by sequence, using the PST1 restriction enzyme. And with this enzyme, we cut the genomic DNA in different places, and we sequence 100 base pairs after the enzyme restriction site. That's a way that we found to have a genomic approach. We could see, we could have a genomic database without having to sequence the whole genome, because it's very expensive and you need to have a very high quality DNA. With those reads, we used IPARED to prospect the SNPs. SNPs are single nucleotide polymorphisms, which means we have mutation in one base pair in a sequence, and that's what we used in this study. And we have about 2,600 SNPs for Nematopsis species 1, 1,200 SNPs for Nematopsis species 2, 1,700 SNPs for Linear Sanguines, and 1,600 SNPs for Pairwise Fst. And it's important to notice that these numbers does not mean that a species has larger or smaller genome. It's just how many sites for the PST1 the genome have. So that's why we have that difference in SNPs. With those SNPs, we use 3 different softwares to look at the population structuration in this species. And we also use 2 other softwares, BaseScan and LEA, to trying to find SNPs that have different frequencies and that might be under selective pressure. I'm going to show to you each of the species separately to be a little bit easier. First, this is the result that we got from structure. Structure tried to show to us the genetic cluster that compose a species or whatever you are looking. And in this case, each of these vertical bars, it's one specimen that we sequenced. And the colors here below are the colors matching the locations here in the map. And the colors in the bars are each genetic cluster that composite that species. It's interesting to notice in this species that the 3 specimens from the northeast, they are here, they have a higher contribution of cluster 1, while the others have more or less equal contribution from cluster 2 and 3. But they also have contributions from the cluster 2 and 3, as the others has contributions from cluster 1, so they are sharing generic material. When you look at the PCA results, PCA is this graph here, and it's trying to show in a graphic space the genetic distance. And what you see here is that all populations are pretty similar. They are close in the same space with some outlier individuals. The colors here are also the same as before, each color for a location. And you see there, even the location in the northeast, Sabiaguaba, it's very similar to the rest. And when you look at the heat map, this is a heat map showing the genetic distance between specimens. And you see the shades of gray are really clear. So they are not that different between each other. With some outliers, there are these guys, they're far in the graph of the PCA graph as well. And when we look at the Pairwise FST, which is this first table here, none of the FST values were significant for these species. They are a little bit higher, 0.3, it's a high FST, but they are not significant. We don't know if that's because we have such a small sample size for this species, or it's because they actually don't have structuration in the populations. The second table is pairwise, average pairwise difference. This first one is the nade distance, and you can see the same pattern as seen in the Pairwise FST. On the top of the matrix, you have the pairwise between populations, the difference between populations, which has the same pattern of the FST. And the diagonal line, you can see the number of pairwise differences inside the population. And it's interesting to notice that we have two populations with higher pairwise differences, but these are the populations that have more specimens. So probably that's why we are seeing this. This third graph, it's an isolation by distance plot. So we are trying to correlate the genetic distance here in the y-axis with the geographic distance in the x-axis. And you see there is some tendency of correlation, but this is not a real correlation. We did not have a significant correlation. And the colors in this graph are trying to show where the dots are. So as close to red you have, it's more dots that you have in the graph. So as you can see, they are very all the graphs, so they are not really going with the line, with the correlation. That's why we don't have a correlation between the two. Looking at Nematopsis species 2, we have 4 genetic clusters, but these 4 genetic clusters doesn't show any pattern. So we probably have connection and gene flow between all populations, but we have these 2 specimens here that have only the genetic cluster 4. They are very different from the rest. And it's interesting because they are in the same location of these other 2 here in Sao Paulo. So why are they so different from the rest, while when you have someone so far away geographically that it's similar to the rest? We think probably because these 2 specimens are, this is a marina that we collect, so they are probably some kind of introduction in the marina. When you look at the PCA, you have the same pattern with most specimens here in the same location, pretty close between each other. And the 2 species, the 2 specimens from Igararese in Sao Paulo, very far away from the rest. The same pattern is shown in the heat maps. When you have those 2 specimens that are very different from the rest, are pretty similar between each other, but they are not that different in distance from the rest. So maybe why they are so far away in the graphs is because they are so similar in comparison to the others that they goes that far. Looking at the FST, we also don't have any significant FST. Once again, we don't know if that's because of our sample size or because they don't have any structuration between populations. We see some high values between Igararese and other locations, but this is probably because of those 2 specimens. They are so different from the rest of the animals. The same pattern is seen when you look at the nade distance and when you look at the pairwise differences. And when you look at the pairwise differences inside the population, you can also see that 2 populations are very diverse. The one that has those 2 individuals that are weird is this one here. And this second population that it's pretty high in pairwise differences, it's interesting because they only have 2 specimens, so they are very different specimens. The isolation by distance plot don't show any correlation between genetic distance and geographic distance as the other. Linneo sanguineus, looking at the structure results, we have actually 7 genetic clusters, which is interesting because we only have 6 sampling sites. So we have more genetic clusters than we have of locations. And that's because most of the populations are composed by more than 1 genetic clusters, as you can see here. All but the population from the northeast, this one in red, have more than 1 cluster, and this one in red doesn't have any contribution from the other cluster. So it's a very isolated population. It is this one here in the northeast. The same pattern of isolation of the population in the Northeast is seen in the PCA right here, but here you have something interesting because we have most specimens here very close, very similar genetically, but you have some specimens from the same locations. They are different and very far away in the graph, both in these two locations in Rio de Janeiro and in this location in Sao Paulo. And the same pattern is seen in the heat map, when you have these very clear squares showing that these guys are very similar. But in the same location, we also have these very dark squares showing how different they are, how distant they are genetically. And looking at the pairwise FST, we now have significant pairwise FST with higher values than we had for Nematopsis. And it's interesting that the one in the Northeast is the most different from the rest. And these two populations, Itaipu and Boa Viagem, are very similar. We almost don't see any difference in FST. And that's probably because these two populations are just a few kilometers apart. The same pattern is seen when you look at NA distance and when you look at the pairwise differences between populations. And when you look within populations, you see that these two, Boa Viagem and Itaipu, are has very high number of pairwise differences. And these populations are these two here, green and blue. The isolation by distance plot this time shows there is correlation between genetic and geographic distance, as you can see also by the colors in the graph and the p-values. So here we start to see some isolation by distance. And for Perinere spontanei, we have only 3 genetic clusters that are forming these populations. One that is unique for this population here in the southeast. One that is shared, this purple one, is shared between these populations in southeast and the populations in the northeast. And the blue one is shared only between populations in the southeast. Looking at the PCA results, we have the same pattern. 3 clusters, one only for the northeast species and 2 northeast populations and 2 populations of the southeast, one with populations only from the southeast, and one that is exclusive from one location. However, when you look at the heat map for distance between individuals, you start to see there's not much difference here. So what these 2 softwares are picking up are very fine scale structurations, because the distance is not between individuals is not that large, but these softwares are picking up this fine scale, this fine structuration that you can see between populations here. Looking at the FST values, we also have some significant FST, especially between that one population that has its own cluster and the rest of them, and between this other population that only has 2 specimens, so might be a sample bias. But if you look at the FST values, you can see that they are way lower than what we were seeing for the Nematopsis and Linnaeus sanguineus. The same pattern is seen for the NA distance and for pairwise differences between populations. And when you look at differences within populations, you see there's a higher difference inside Araçá, but this is the population that we have most animals from. So it might be just because we have more animals. Or maybe because we have those individuals that are so different that you can see in the PCA. Looking at the isolation by distance plot, you can see there is correlation between geographic and genetic distance. It's lower than the one that we find for Linnaeus sanguineus, but it is there. So what all of this indicate to us? We could see that Nematopsis populations of both species seem to be very well connected, and Linnaeus sanguineus and Linnaeus spontanei show signs of isolation by distance besides the admixture that we could find. And as I said before, the pelagic larval duration, it's correlated with the isolation by distance. So as long as you have a larval duration, you have a smaller isolation by distance, as you can see here in these two graphs from two different papers that were working with many different species, marine species all. Of course, we have some outliers, as you can see in the graphs, but as a general rule, this is true. And what this tells us about the Nematopsis development strategy, as Lana was saying before, we have three different types of Haplotype larvae. The Pelidium larva that is common for the Pelidiophora, a planuliform larva that is common for Paleonemortes, and it's known to feed. Here you can see with a bivalve inside the gut. And the planuliform larva that is known for the Haplonemordians. And we already have some indication that Haplonemordians are actually feeding larvae, but most literature, what you see is that they are probably lithotrophic. They don't have any special characteristic that will give them the ability of feeding the plankton. But when you look at some Nematopsis that we found with eggs, we could see that the eggs are not that big. They are not more than 100 microns, which is expected for a lithotrophic larva. So this is one more characteristic that is telling us that they are probably not lithotrophic, which made me reach Svetlana and ask and talk about if I could go to Oregon and study how these larvae live and how they develop and what they eat. And we see that in Planktonema viride actually do feed in the plankton. They feed upon Nauplii, barnacle Nauplii, and barnacle cyprids. And in Planktonema viride, it's a very similar ecologically to Nematopsis. They have the same morphological characteristics. They feed upon the same thing, acorn barnacles. So we think that the larva of a Nematopsis will be pretty similar to the larva of Planktonema viride. So we are probably looking at two species that actually have a feeding larva, and at least for Planktonema, these larvae can take up to 6 months to reach metamorphosis, so they would be able to disperse a lot and connect the species. Okay, so yes, the larva is very important. It is dispersing, but look, what about the other two species, Venus sanguineus and Pairwise Fst? We need to remember that besides the larval dispersal, we have a lot of other different things that will influence the population connection and how we see this population connection. And that's what this figure is showing. But I'd like to pay attention to the barriers to dispersal, because for Pairwise Fst, we do have low values of FST, so you can see that they are well connected, but we also can see that there are something making these populations harder to connect, because you have a population that is pretty close geographically from the rest in the southeast, but it's genetically more similar to the populations in the northeast. And why is that? It's important also to notice that there is another study that tried to see how the population connectivity of these species in Brazil occurs, and they used mitochondrial DNA, CO1 and 16S DNA to try to describe the population connection. And here you can see two haplotype networks. This first one is for CO1, the second one is for 16S. And as you can see, they don't find the same pattern. They don't find a differentiation between haplotypes. Each color here is a state in Brazil, both from the southeast, the south, and northeast. And they don't find that differentiation between populations. And that's what is so interesting about genomic markers, because you can find these finer structurations that some markers will not show you. Even if you use some nuclear markers, it's a smaller sample that you have from that genome, and you sometimes will not find these finer structurations that we could find with genome markers. So, okay, Brazil, have a major current, that's the Brazil current here. And this current, it's probably what is connecting all these populations. So what is happening here in this area that it's not, that it is impacting these populations? During the summer months in Brazil, we have some wind that blows from the mainland through to the ocean in this region here of Cape Frio in Rio de Janeiro. It's in the southeast. And when these winds come in the summer, they are pretty strong and they cause an upwelling of cold water in this region, which is known to be a barrier for some larvae and for some animals. And what we think is this is also a barrier for these larvae to reach. They are coming from the south, from the northeast, reach the south. So that's what we are picking up in these analyses. And what about Linnaeus sanguineus? These species, first we have a very isolated population here in the northeast. And once again, ocean currents are probably responsible for that, because we have the south equatorial current that comes to Brazil and reaches the northeast and splits into the southwards Brazil current and the northwards North Brazil current. And probably what is happening at this is a superficial current, and probably this is where these larvae or these animals are dispersing. So they cannot go, the species that the specimens that are here cannot go southwards, and then you have this isolation. Of course, this is a median of the current. This current is motile. It will change what it's reaching, can sometimes go northwards and southwards. So you still have some animals coming southwards, but this is probably what is causing the isolation here. And what about these so different animals that are in the same locations as the others, but are so different in genetics? As I said before, this species is known to have asexual reproduction. So what we think is that we have clonal individuals that are very similar here between each other, but they are very different from the rest, because when you have asexual reproduction, you will have the individuals sharing somatic mutations, because once you only need to break a piece to make a new individual, you see the somatic mutations being shared in that clonal population. But what about the rest here? They are so similar in different locations. They are quite distant. This paper also shows, also says that the FIS, it's NAF statistics, this value here. This is statistics try to calculate the inbreeding inside a population. And when you have higher values of FIS, you probably are seeing inbreeding inside a population. When you have lower values, you don't have much inbreeding. And negative values, according to this paper, are indicative of asexual reproduction. But here in our population, we have FIS, a positive FIS, and if you look here in this graph, which is the expected heterozygosity versus the observed heterozygosity, you see there is a lot of homozygosity in the population. So they are probably, these guys here are sharing gametes and they are reproducing sexually, and that's why we have this pattern of isolation by distance and shared of genetic material. Okay, so we now know that both Nematopsis species have a high population connection, meaning that they probably have this long-lived larva as Inflectonema virid and other hoplonemeridians that we don't know yet. Linnaeus sanguineus populations present signs of admixture, showing that they probably have sexual reproduction here in Brazil, which is interesting because no one ever saw these specimens or any Linnaeus sanguineus with gametes, but looks like they are sharing gametes because that's the only way that they could share genetic information. And Pairwise spontaneous populations, they are very well connected, but they seem to respond to that ecological barrier that's happening in the southeast. So we now know that different reproduction types do influence the connectivity in these species. However, other factors such as the ocean currents and the environment also have an important impact. And having that in mind, we started to look to those SNPs and trying to find the ones that might be under selective pressure and that might give us some answers of how these populations are going to respond to a change in the environment with the whole climate change. So at first, we used Basecamp, which is a software that's trying to find FST outliers and does not correlate with any environmental variable. So we use this for all 4 species. For the 2 Nematopsis species, we could not find any SNPs that are candidates to be under selection, but this is probably because we don't have a big sample size, especially because this software uses the FST of a population to find the outliers. And once you have some populations with only 1 or 2 specimens, they cannot find these outliers very well. But if you look to Sanguines and Pairwise spontaneous, we actually find a lot of SNPs that are candidates to be under selection, and show there are some selective pressures happening in these populations. And it's interesting because for Sanguines, here we have a lot of SNPs that are under positive selection, but we also have some that are under purified selection, while for Pairwise spontaneous, we only find SNPs that are under positive selection, candidates to be. We cannot be sure that they are under selection, but they are candidates because of these outlier values of FST. We also try to correlate with the ecological variables, and how we did that. We use the LEA package using the LFMM algorithm to find the variables, the environmental variables that best explain the distribution of a species. And using those variables, we try to correlate with the frequency of SNPs. And what you're seeing here are Manhattan plots with SNPs. So each of these gray dots, it's a SNP that it's not correlated, doesn't seem to be correlated with such environmental variable. And each red dot, it's a SNP that seems to be correlated with the environmental variable. So here you can see that for this species, Nematopsis species 1, we have that the chlorophyll, the mean chlorophyll, and the salinity of the sea surface are the most important variables shaping those frequencies. We also have precipitation and temperature also as factors that are shaping these frequencies of these SNPs. For Nematopsis species 2, we could not find any SNPs. They are correlated with environmental variables, but this is probably because of the sample size. Once again, we only had 16 specimens, so probably that's why. For Sanguines, we have only 7 variables that seem to be correlated that seems to explain the distribution of the species. And the ones that seems to explain better, they are more correlated with the SNPs, are the temperature, the precipitation, and the dissolved oxygen. And it's interesting because that the temperature it's here because this species is known to like better cold environments, but we also can find them in the northeast part of Brazil, which is really hot. So this is really interesting to see. And for Pairwise spontaneous, we had a lot of SNPs correlated with the 8 variables that we could find that better explain the distribution. And these are mostly correlated with temperature. It's the first one here, chlorophyll, salinity, precipitation, and temperature of the air. So now we are trying to find the SNPs and trying to see if they are in coding regions of the genome. And if they do, what are these coding regions coding for? What kind of genes are they correlated with? And if they are correlated with some kind of structured genes or if it's something from the response of the environment. We still don't know. We are still doing that work. I still have a year of PhD, and that's what I'm doing right now to understand better what is shaping these populations and how they will respond to a changing environment. And with that, I would like to thank you, my lab mates. They are here. My supervisor, Sonia Andrade, my unofficial co-supervisor, John Norenberg, Cynthia Santos, my co-supervisor, Svetlana Maslakova, who welcomed me so well at OIMB and helped me to understand better the Haplotype larvae. Elena Matthews, that gave me support in the sampling in the Northeast. Paulo Pacelli, who helped me sampling all the places in Brazil. My funding agencies, OIMB, my Institute of Biosciences where I work, and Sebimar, who helped me with sampling in the Southeast. With that, I'll take questions.
Thank you very much, Cecili. That was really wonderful. I'd like to ask everybody to give you a round of applause. Show our appreciation. And we'll now open it up for questions. Looks like Svetlana has a question.
Yeah, so thank you, Cecili. It was really cool to see all that work you did in Brazil on those species that I'm not familiar with, except for Sanguines. And it's really cool how the work that you did with George and myself at OIMB ties in with your genomics work in Brazil. It's such a wonderful example of I don't know how to put it, pieces of the puzzle coming together. I'm really glad that it worked out that way. And I just wanted to mention once again George's role in this. So George is actually the one who, for the first time ever, observed directly the feeding by Haplotype larvae on planktonic crustaceans. I had some suspicions about it based on my observations in larval barcoding, but George was the one who actually first saw it, and Cecili was the first one who actually, using that information, was able to raise them. So that was, yeah, great collaboration. So I have a question that's not especially relevant to the life histories or the dispersal potential of these species, but something that I'm curious about, have been for the last few years, working with biodiversity of ribbon worms in various parts of the world, here in Oregon, as well as in the Caribbean, in Panama, and discovering huge numbers of cryptic species. As you know, we barcode them and find that what we thought was one species is 2, 3, 4, 5, 6, sometimes more. And they all co-occur. And so that seems to be also the case with your 2 Nematopsis species from Brazil.
Yes.
They co-occur in the same location and are close relatives of each other, presumably sister species.
Probably.
Yeah, and they have similar, presumably, feeding habits. They both feed on barnacles, as you say. So what gives? How do they maintain reproductive isolation? How do they? Do you have any comment on that?
I actually don't know how they maintain isolation, but especially because when we're looking at mitochondrial DNA, we found some exchange between the 2 species. So they actually probably have some kind of exchange, maybe some ancient exchange of gametes, but we still can see the signs looking at mitochondrial DNA. Looking at genomic DNA, we could not find much of genetic sharing between those 2. And I don't know how they do that, because they, besides live together, we could collect them in the same place. They also have the same period of reproduction. So I have no idea how they do it. Probably some kind of chemic cue or something that helps in the isolation, but I still don't know how that happens. I really want to try to find out how, but I still don't know.
I think Richard has a question.
Cecili, thank you for the very nice seminar. You lost me very quickly into the genomics part. But, I can't help but wondering, related to Svetlana's question, I have two questions. Related to Svetlana's, how distant are the two Nemertians, the sister species, in terms of their CO1 differences?
I don't remember the percentage, but they are quite different, especially in the genomics. Because when we thought it was only one species, we're trying to have all those SNPs together, and we could not get the same SNPs from the two species. So they are very different genetically. And CO1 distance, I believe it's something about 7 to 10% of the difference.
I mean, if they're, do you think they're sisters, true sisters, and that they share a common ancestor?
I think they share a common ancestor.
A single common ancestor?
But not, I don't know if it's a single one, because you have more than 2 Nematopsis species that are lookalike. Here in Brazil, we have at least 3. I worked only with 2 because it's the ones that I have more locations that I could find them. But I have 3 here in Brazil and maybe more in the North America. So I don't know if they are true.
The distribution was striking, and it looked as if the Amazon could have had some historical influence on their distinction. That's where I was headed with that. But is there any way to look at the genomes and ask if they're old or new changes? And I mean, it seems like while you're comparing species with different modes of reproduction, you're also comparing species with long histories, as you pointed out. And I just was trying to get a feeling for whether some of the patterns were related to recent speciation as opposed to just long-term persistence as a species. And that's a really harebrained question, but I was wondering it while you were talking.
With my data, we can't, because I do have genomic data, but it's not that much, because I only have 1,200 SNPs of one of those species. But if you have a different kind of genomic data, yes, you can try to find that. You have some software that can do that.
Okay, thank you very much.
Sure, no problem.
George?
Hi, Cecili.
Hello.
Nice to hear your talk. I was curious because, I mean, in part, I'm naive about these things. The case in which mitochondrial markers don't show you any significant differentiation, but your SNPs do, right? And your SNPs are not, I mean, it's not like this is microsatellites, right? You don't expect them to be necessarily any more sensitive to intraspecific variation, right? Or do you? I guess, so the question is, how often, do you know how often people find that if they've sampled populations with mitochondrial markers and found that they are more homogeneous than if they take the kind of approach you did?
So that's mostly because of the nature of the marker, because with mitochondrial DNA, you have mostly information about the maternal lineage, and these are usually more old differences that you can find. With SNPs, you get mostly nuclear DNA, and you have more markers. It's like you're working with more information. That's why you can have this fine scale information from them. And since it's nuclear, they have more recent change on them. You can get this more recent differentiation in the populations. That's why you find with SNPs what we don't find with mitochondria.
I guess I'm wondering if something like that is not just about the numbers, but about the relevance of recombination to either sustaining the accumulation of neutral variants or alternative. I mean, there are alternatives. And so I guess I wondered how often you're aware that people find, I mean, it sounds like you're saying that's sort of an expected outcome just because you have more markers.
Not just because you have more. I mean, more, it's better because you have more information, but it's because of the time of the change. because you have different time, exactly what we're saying, that you have different times for the recombination to happen between mitochondrial DNA that doesn't recombinate much and the nuclear DNA. So it's also about the timing that you're looking at. You have a different time to look at. And it's common to find different things between mitochondria and nuclear and SNPs. It's pretty common when you look at population level.
Ernie Manders, did you have a question?
Yes. Cecilia, you have combined the best of both worlds. You have a field collection career combined with a sophisticated molecular biology career, and that's just wonderful. My question is, David Baltimore, Nobel laureate for reverse transcriptase, past president of Caltech, said, biology has become an information science. And as I watched your wonderful presentation, I thought to myself, how did she keep track of all that data? Could you just describe for us the information systems that you use to gather, collate, and then display all that data?
So I used, at the sequencing level, you have different barcodes for different specimens. So that's how we keep track of each of the specimens. And at the bioinformatic level, we use different softwares that can prospect the SNPs, and the ones that we use those SNPs to gather information. And it's actually not that hard, because all these softwares are constructed in a way that you can keep track of what you're doing pretty easily. You just need to have some bioinformatic skills. Once you have that, it's not that hard. Once you have the SNPs, they are all very organized between individuals, and you can work with each individual. And from that, you just use some bioinformatic tools to have those results. It's really not that hard.
I'll have to take your word for it. Thank you.
Sure.
Are there any students with questions? Here's your opportunity. Okay. Well, Cecili, thank you very, very much. That was beautiful.
Thank you.
And we really appreciate your participation here. We'll give you one last round of applause and then let you go. Thank you very much.
Thank you for having me. I was really missing the Oregon community. It was really nice to be here, even in Brazil.